now we're going to see how they become activated. So as long as they are immature, they don't express co-stimulatory signals and MHC. But when they capture the bacteria, so I'll draw a bacteria here, with the help of complement toll-like receptor, FC receptor, they engulf it, break them down into small pieces, and now they will present it with the help of MHC and with the help of co-stimulatory signal like CD80 and 86. And they will move towards these afferent lymphatics and go to the lymph nodes. So these are the follicles, and they will come here in the parafollicular region. So by the time they reach the lymph nodes, now they are called as mature dendritic cells. Better on mature dendritic cells, they have got MHC expression on them, and they have got CD80 and CD86. These are the receptors that makes it mature. So when they are immature, they don't have co-stimulatory, they don't have MHC, they just have complement receptor, toll-like and FC receptor. They help in phagocytosis, and once they are inside the cell, then they will become activated and they will slowly start moving towards the lymph node, and they will express MHC, then CD80, 86, and now they are called as mature dendritic cell. On the other hand, the macrophages, they also have C-reactive complement receptor, FC gamma receptor. They will have toll-like receptors, and here is the bacteria now. They will recognize it, and when they recognize it with the help of all these receptors, they will start expressing as well co-stimulatory signals, and they will produce lot of things. So I'll just draw a bigger macrophage here. Inside the macrophage, you can see this is a phagosome and this is a lysosome. The phagosome, when they engulf the bacteria inside, now they start to become activated. And one of the things they will do is, in their phagosome membrane, there is an enzyme which is called as NADPH oxidase, and that will help in the production of H2O2, hydrogen peroxide. And also, so these are reactive oxygen intermediates, and also produce nitric oxide, and these are called as reactive nitrogen intermediates. When they are produced, I will now here draw another receptor on the macrophage. It is called as interferon gamma receptor. I told you that the T cells, they can activate. So there are two phases of activation. Phases. One phase is when toll-like receptor or the complement receptor, when they bind, it will go in phase 1. It will be like 60% activation. Then phase 2 is when interferon gamma, they bind, then the macrophage will become 100% killing machine. And by the second phase, what will happen? This hydrogen peroxide and nitric oxide, with the help of myeloperoxidases, they will convert into hypochlorite and peroxynitrite. So these are converted into hypochlorite and they are converted into peroxynitrite. This is 100% activation. This is 60% activation. This first phase is under the influence of TLR, C protein, and the second phase is under the activation of interferon gamma, and they will convert these hydrogen peroxide and nitric oxide into hypochlorite and peroxynitrite. Now the macrophage can kill any pathogens, in particular, for example, mycobacterium, which can resist other, but they cannot even resist these very strong killing chemicals that are produced.